This lecture is about neurotransmitters. We started talking about neurotransmitters in the last lecture, but I thought we should review some of the information and then go on from there. And so a neurotransmitter is a chemical that transmits information from one neuron to another. So you have the action potential coming down the axon of the presynaptic neuron. And then that action potential somehow has to get to the next neuron. And so it's the neurotransmitter that sort of takes the information from the presynaptic neuron, crosses through the synaptic cleft, and then transmits that information to the postsynaptic neuron. And we talked about acetylcholine, which transmits information from a somatic motor neuron to a muscle cell. And acetylcholine also transmits information within the central nervous system and in the autonomic nervous system. And we'll be taking a look at that in upcoming lectures. And then there are many other neurotransmitters that we will be talking about in this lecture. But just going back to acetylcholine, it can have very different effects on different tissues. And in some cases, it can be excitatory, and in other cases, inhibitory, depending on the organ involved. And so you might think, well, gee, how could that happen? And so this happens because acetylcholine can bind to two types of receptors. And acetylcholine receptors are called cholinergic. And you have the nicotinic acetylcholine receptors. And they are named nicotinic because they can be stimulated by nicotine. These are found on the motor end plate of skeletal muscle cells, in autonomic ganglia, and in some parts of the central nervous system. And there you have a picture of acetylcholine and nicotine. But then acetylcholine can have other effects because it binds to what are called muscarinic acetylcholine receptors. They're called muscarinic acetylcholine receptors because they bind to muscarine, which is from poisonous mushrooms, which you can see in that picture. And muscarinic acetylcholine receptors are found in the central nervous system and also on the plasma membrane of smooth muscle and cardiac muscle and glands that are innervated by autonomic motor neurons. We'll be talking about them a lot when we talk about the autonomic nervous system. Let's review on the next slide how acetylcholine works. And so this slide is about acetylcholine binding to the nicotinic acetylcholine receptor. Okay, so remember that's the one that is found on the motor end plate of skeletal muscle cells, but also in the autonomic nervous system and central nervous system. And so in this slide, what we can see is that we have the channel closed, okay, and the channel is shown in purple, and nothing can get in because it's closed. But when acetylcholine binds, which is shown in green, two acetylcholine molecules bind to this receptor, which is the nicotinic acetylcholine receptor, then the channel becomes open and permits the diffusion of sodium ion into the muscle cell. And you can also see that some potassium leaves, but primarily what is important is that sodium is entering the muscle cell or postsynaptic neuron. Okay, so in this case, acetylcholine binds to a receptor that's also the ion channel. But the muscarinic acetylcholine receptor is not an ion channel. And so the muscarinic acetylcholine channel makes use of what are called G proteins. And G proteins won the Nobel Prize in 1994 in physiology or medicine, was awarded to Gilman and Rodbell, and they discovered the G proteins, but then in 2012, the Nobel Prize was given in chemistry for studying the function of G proteins to Robert Lefkowitz and Brian Kobilka, shown in these pictures. And so let's take a look at the muscarinic acetylcholine receptor. And so in this case, the receptor is not the ion channel. Acetylcholine binds to the receptor. Again, it's shown here in green. And then it binds to the receptor. But the receptor is associated with G proteins. And these G proteins are the alpha, beta, and gamma G proteins. And when acetylcholine binds the receptor, the beta and gamma G protein subunits dissociate and they move through the membrane and they bind to the potassium ion channel. The potassium ion channel then opens and potassium goes out of the cell. And so in this case, we remember that if potassium is going out of the cell is becoming more negative. And so this is going to initiate an IPSP, but also in other tissues, the binding of G proteins could close the potassium ion channel and initiate an EPSP. Okay, so in the case of IPSPs, this is the effect of acetylcholine on the heart. Acetylcholine from the parasympathetic nervous system binds to the acetylcholine receptor on heart muscle tissue, creates an IPSP, and the heart rate slows down. And then on smooth muscle, though, potassium channels close when acetylcholine binds. That creates an EPSP and a contraction of the smooth muscle. So that's then excitatory. We also talked about how drugs are based on a knowledge of how receptors work. For example, agonists are drugs that can stimulate a receptor, such as nicotine for the nicotinic acetylcholine receptors and muscarine for the muscarinic acetylcholine receptors. And then antagonists are drugs that inhibit a receptor, such as atropine, which is an antagonist for a muscarinic receptor. And, one use is during surgery, it will decrease saliva production from a patient. Atropine is from this plant, Atropa belladonna. And also curare from plants is an antagonist for nicotinic receptors and is used as a muscle relaxant. And here's a picture of the curare vine from where curare is isolated. And then after acetylcholine has its effect on the postsynaptic cell, it is then degraded by acetylcholinesterase. Okay, so that's the enzyme that inactivates acetylcholine activity after it binds to the receptor. Okay, and there's a picture of it. It hydrolyzes acetylcholine into acetate and choline, which are then taken back into the presynaptic cell, and they can be reused. And so here's the chemical reaction. You don't need to know the molecular formulas, but just realize that acetylcholine is broken down into acetic acid and choline. And these enzymes are actually embedded in the membrane of the postsynaptic cell. And so in this picture, you see acetylcholine going into the enzyme acetylcholinesterase. Let's look at this one here, actually. And acetylcholine goes in. They have it changing a color because it's then going to be broken down into acetate and choline, which are then taken back into the presynaptic cell where they can be used for the synthesis of more acetylcholine. So I'd like to go on and talk about more neurotransmitters, for example, the monoamines. And the monoamines are neurotransmitters that have amines as part of their chemical structure, and they have a single amine, 1. And so monoamine neurotransmitters are derived from amino acids. And here we have a picture of the amino acid tyrosine. And so the general structure of an amino acid, if I could circle this here. Okay, let's just go like this. Okay, so you have the amine, and you have amino, and you have the acid. And then you have this group down here, which is called the R group. And I know my students are aware that there are 20 amino acids, and they all have a different R group. And so this is the R group for tyrosine. And the catecholamines are neurotransmitters that are derived from tyrosine. This ring is known as a catechol ring. Dopamine, norepinephrine, and epinephrine are catecholamines. Norepinephrine, sometimes called noradrenaline, epinephrine, sometimes called adrenaline, and they bind to adrenergic receptors, and you can see and hear that adreno in the word. Norepinephrine is a neurotransmitter and a hormone, but epinephrine is primarily a hormone. Other monoamines are serotonin, derived from a different amino acid, tryptophan, or histamine, derived from the amino acid histidine. So let's talk about norepinephrine. Okay, norepinephrine is used in both the central nervous system and peripheral nervous system. And in the sympathetic nervous system, remember that's sort of your fight or flight nervous system. And norepinephrine is going to be used on smooth muscle, cardiac muscle, and glands. I mean, when I think about norepinephrine, the first thing I think about is this is the neurotransmitter that is activated when you exercise and increases heart rate. Okay, so, but it does a lot of other things that we'll talk about. Used by neurons of the central nervous system in brain regions associated with arousal, for example. And amphetamines work by stimulating norepinephrine in the brain. And so let's just look at a diagram of norepinephrine as it is released from the presynaptic neuron. Okay, and so realize that it is a tyrosine derivative, and so it actually goes through this metabolic pathway where tyrosine is converted to dopa, which is converted to dopamine, which is then converted to norepinephrine, which is then stored in synaptic vesicles. You can see these synaptic vesicles. And when the action potential rolls down the axon and depolarizes the terminal bouton, calcium ion channels open up, and calcium enters the presynaptic neuron, which leads to exocytosis of the synaptic vesicles, and the neurotransmitter then enters the synaptic cleft. Okay, so we're talking about norepinephrine here. And then it binds to its receptor on the postsynaptic cell. After norepinephrine does its job, it's actually taken back directly into the presynaptic cell and inactivated by an enzyme, which it says MAO here, or monoamine oxidase. Okay, so monoamine oxidase. So actually norepinephrine is broken down after it gets taken back into the presynaptic cell. Some people are on antidepressants like monoamine oxidase inhibitors because norepinephrine can actually make you feel more peppy. So its lingering presence in the synapse may act as an antidepressant. So you might hear about monoamine oxidase inhibitors. Okay, so monoamines use G-protein coupled channels. We just went over that for the muscarinic acetylcholine receptor, But let's look at what happens in the case of norepinephrine. So this is a beta adrenergic receptor. And in this slide, what you're seeing is norepinephrine binds to its receptor, and the receptor is associated with these G proteins. And the binding of the receptor causes the dissociation of the alpha subunit, which then travels through the cell membrane and binds to the enzyme adenylate cyclase and activates adenylate cyclase. Adenylate cyclase is an enzyme that converts ATP to cyclic AMP, and you sometimes see cyclic AMP written as CAMP. And cyclic AMP is then a second messenger, which activates a lot of different enzymes. In this case, what they're showing is that an inactive protein kinase becomes an active protein kinase. An active protein kinase, its function is to add phosphate to proteins, so it phosphorylates proteins. And this active protein kinase also opens ion channels, and so then ions can flow into the cell and create a depolarization event. And sympathetic neurons of the peripheral nervous system use norepinephrine as their neurotransmitter. So we talked about sympathetic fight or flight response, action of a peripheral neuron on the heart, let's say, which will then speed up heart rate. Let's switch gears and talk about serotonin as a neurotransmitter. Okay, and this is used by neurons in the Raphe nuclei, which is in the middle region of the brainstem, which I got this picture off the internet. And so these are called serotonergic neurons because they produce serotonin. And serotonin has been implicated in mood, behavior, appetite, and circulation, cerebral circulation. And LSD and other hallucinogens may actually be agonists of serotonin. Serotonin is sort of a neurotransmitter that kind of makes you feel that you are in a steady state. And so some antidepressants make use of our knowledge of serotonin, which is that the longer it lingers in the synapse, the better a person feels. And so there's a whole class of SSRIs, or serotonin-specific reuptake inhibitors, that are used to treat depression, such as Prozac, Paxil, and Zoloft, for example. And there are many different receptors that are known for serotonin. So it has kind of diverse functions, and many different drugs are going to target these receptors. We talked about depression, anxiety, appetite control, even migraine headaches can be treated by SSRIs. And let's talk about dopamine as a neurotransmitter. Neurons that use dopamine are called dopaminergic neurons, and they're highly concentrated in the midbrain area, which we can see here in two main areas: the nigrostriatal dopamine system, which is involved in motor control, which I put a blue box around. And the nigrostriatal neurons are coming from the substantia nigra, which can be seen here. And they are going to the basal ganglia, to areas like, showing these neurons in blue here, the caudate nucleus and the putamen. Okay, and these areas are involved in motor control. And then there is the mesolimbic dopamine system, which I put a red box around, and the axons are coming from the ventral tegmental area and are going to the nucleus accumbens and areas of the prefrontal cortex. And the mesolimbic dopamine system is involved in emotional reward. So let's go back and talk about the nigrostriatal dopamine system involved in motor control. And that's shown in the next slide. And so this area is associated with Parkinson's disease, so degeneration of these neurons makes sense. They're involved in motor control, and because we know that they are stimulated by dopamine, their patients with Parkinson's disease are treated with L-DOPA, which is a form of dopamine that it's a precursor to dopamine that is able to cross the blood-brain barrier, and also monoamine oxidase inhibitors, which would keep dopamine in the synapse for a longer length of time. Okay, and so this is going to activate the substantia nigra. And then you have the mesolimbic dopamine system, which is for emotional reward. And schizophrenia is associated with too much dopamine. And so dopamine antagonists are used to treat schizophrenia. And so this area is from the ventral tegmental area to the prefrontal cortex. It also activates the limbic system, and so can be associated with addictions to nicotine, alcohol, and other drugs. That the limbic system is the part of the brain which consists of a lot of different parts that have been found to be involved in emotions. Okay, and then cocaine has been found to affect many different neurotransmitters, and one of the reasons it's abused is because it elevates your energy and mood. And all of these neurotransmitters that we've been talking about, like dopamine and serotonin, also norepinephrine, are all mood-enhancing neurotransmitters. And so cocaine is able to cross the blood-brain barrier. And in the mesolimbic reward system, it acts as a triple reuptake inhibitor, blocks reuptake of dopamine, serotonin, and norepinephrine. And so all these neurotransmitters are staying in the synapse for a long period of time. And you can imagine why people become addicted to cocaine. But the danger is it's blocking sodium ion channels and has all sorts of effects that are not about making a person feel good, such as raising heart rate and blood pressure. It also has been found to increase risk of stroke and seizure, among other things. So let's move on and talk about some other neurotransmitters. For example, amino acids. So we were talking about amino acid derivatives, okay, but now we're talking about amino acids themselves. And so main excitatory neurotransmitters are glutamate and aspartate. Glutamate is the major excitatory neurotransmitter in the central nervous system. It produces EPSPs in 80% of synapses in the cortex and uses major use of energy in the brain. It's also been found to be involved in memory, which we will talk about. And astrocytes are known to take glutamate from the synaptic cleft to increase glucose uptake and increase blood flow by vasodilation. Okay, so the glutamate receptors are receptors that serve as ion channels, and they're named according to molecules they bind, which we've seen that to be a pattern. NMDA receptors, AMPA receptors, and kainate receptors all bind to those respective chemicals. But the NMDA and AMPA receptors work together in the formation and storage of memories. And then you have the inhibitory neurotransmitters, for example, glycine, which produces IPSPs. And so how does an IPSP get produced? Well, one way is the binding of glycine opens chloride ion channels. And so this causes an influx of negative charge into the postsynaptic cell. And if a cell becomes more negative, then it's harder to reach a threshold. One place this has been found to be important is in spinal cord regulation of skeletal muscle movement. Okay, so you have your antagonistic muscles. And so, for example, if you are contracting your triceps, then the bicep muscle relaxes. Well, how does that happen? Well, this neurotransmitter glycine, which prevents action potential, basically, it's inhibitory, will relax the bicep while the tricep muscle contracts. And glycine also has other effects. For example, because it's inhibitory, there's a picture of it there. Actually, glycine is the most simple amino acid. Its R group is only a hydrogen ion, or hydrogen atom. And so because it's inhibitory, it's been found to be involved in relaxation of the diaphragm muscle, which is required for breathing. And the poison strychnine blocks its receptors, which is why people die of strychnine poisoning due to death by asphyxiation. And then GABA is another inhibitory neurotransmitter. There's a picture of it there, also known as gamma-aminobutyric acid. This is the most common neurotransmitter in the brain, used by about a third of the brain's neurons. Also inhibitory by opening chloride ion channels. And this one is involved in motor control. So we're talking about movement here. And Huntington's disease has been found to be associated with a degeneration of GABA-secreting neurons in the cerebellum. On the next slide, we're going to look at the mechanism of action of GABA. And so in this case, you can see GABA at the top of the slide, and the channel, GABA receptor, is an ion channel, but the channel is closed until GABA binds to the channel. And once GABA binds to the channel opens, and these green chloride ions start to enter the cell. And the cell goes down in charge, creating a hyperpolarization or an IPSP. In addition to amino acids, polypeptides can be used as neurotransmitters. These are also called neuropeptides. A polypeptide is a protein. It's generally thought, it's sort of considered to be a fairly short protein of less than 100 amino acids. And these are used as hormones and also paracrine regulators, but they're also used in the brain as neurotransmitters. And so when you hear about them later in the course, they're doing other things in the body besides acting in the nervous system. For example, cholecystokinin, which is released in the intestine and regulates the flow of bile in the intestine, also acts as a neurotransmitter involved in a feeling of fullness or satiety after a meal. And then substance P, P equals pain. That's how this neurotransmitter was named. Mediates sensation of pain in certain neuropathways in the brain. And then in another class of neurotransmitters, you have the endogenous opioids. Okay, endogenous meaning your body makes them, and opioids meaning that they are sort of like opium. Okay, and the opioid receptors are so named because they bind to drugs like opium and morphine, which then results in pain relief. And there's a picture of the Asian opium poppy plant, Papaver somniferum, from which opium is isolated. But endogenous opioids are polypeptides that are produced by the brain and pituitary gland. And for example, enkephalins, beta-endorphin, and dynorphin are examples of endogenous opioids. They produce euphoria, so they're involved in making you feel rewarded. And they are also, for example, endorphins found to increase after exercise and are related to a feeling of well-being as a result of exercise. Another neuropeptide is neuropeptide Y, and this is the most abundant neuropeptide in the brain, playing a role in stress response, circadian rhythms, and cardiovascular control. This inhibits the release of glutamate in the hippocampus. And so too much glutamate can cause, it's, the excitatory neurotransmitter we talked about, can cause convulsions. It can cause something called excitotoxicity, and neuropeptide Y has been found to be an inhibitor of high glutamate levels. Neuropeptide Y is also involved in the hunger response. In addition to neuropeptides, you have the endocannabinoids. Okay, these are neurotransmitters that bind to the same receptors that bind to THC found in marijuana. Okay, and so they have effects that are similar to that of THC, tetrahydrocannabinol. And these are short fatty acids that are produced in the dendrites and cell bodies, and they are released from the plasma membrane directly because they're fatty acids, so they diffuse right through the membrane. And so being that they're coming out of the dendrite and cell body, they actually go backwards into the presynaptic neuron. And so because of that, they're called retrograde neurotransmitters. And they're found to be inhibitory of neurotransmitter release from the presynaptic neuron. And these have been found to actually stimulate memory. But your textbook makes the point that it is the endogenous endocannabinoids rather than THC that stimulates memory. But that exogenous use of THC has been found to actually decrease IQ points significantly. Nitric oxide is another neurotransmitter, not to be confused with nitrous oxide or laughing gas. Nitric oxide is produced from the amino acid arginine in the central and peripheral nervous systems. And it is going to simply diffuse out of the presynaptic cell. And then it will diffuse into the target cell where it can activate the production of cyclic GMP as a second messenger. Okay, and then cyclic GMP causes blood vessel dilation. The drug Viagra works by stimulating the action of nitric oxide because it prevents the breakdown of cyclic GMP. And nitric oxide also has been found in helping to kill bacteria. It also acts as a retrograde neurotransmitter. So it can go from the postsynaptic to the presynaptic cell, or from the presynaptic to the postsynaptic cell. Another gas that's a neurotransmitter is carbon monoxide. And this also activates the production of cyclic GMP in target cells. Okay, and it's been found to be involved in sensory adaptation in olfactory neurons. So these are your smell neurons. And sensory adaptation is this phenomenon where, if you walk into a room and somebody's got a turkey in the oven, you say, oh, wow, it smells really great. And then after a few minutes, you no longer smell it because you've adapted to the sense. Okay, and carbon monoxide has been found to be involved in that response. ATP and adenosine can also be used as neurotransmitters. In this case, they can be co-transmitters that are released in vesicles along with a neurotransmitter. And so they are purines, and so therefore the receptors that they bind to are called purinergic. There's P1 receptor for ATP, P2 receptor for adenosine, and they can act like neurotransmitters with norepinephrine to stimulate constriction of blood vessels, or with acetylcholine if in the stimulation of intestinal contraction. So smooth muscle contraction. And they can also be released by non-neural cells and act as paracrine regulators. So they can be released from the blood vessel endothelium, and they have a role in blood clotting, but they're also involved in taste and in pain. When we think about the action of neurotransmitters, we have to consider this phenomenon of synaptic integration. So we're really talking about, well, how does a neuron respond to a neurotransmitter? And one of the things you have to keep in mind is the organization of neurons with respect to other neurons. And so, for example, you have this phenomenon of convergence. And you can see convergence happening in this picture. And basically, the neuron receives input from several neurons. So in this picture, you have neuron 3 is receiving input from neurons 1 and 2. So you have impulses that represent information from different types of sensory receptors, depending on what triggered neuron 1 and what triggered neuron 2. And so this allows the nervous system to collect, process, and respond to all sorts of information, and makes it possible for a neuron to sum impulses from different sources. And so realize that IPSPs and EPSPs are then summed by the neuron that is receiving them from different neurons. Okay, so this is the phenomenon of convergence. And it also works in the opposite direction, called divergence, which is that one neuron sends impulses to several neurons. So neuron 4, in this case, is sending impulses to neurons 5 and 6. And the role of this is that it could actually amplify an impulse. Okay, so an impulse from a single neuron in the central nervous system might activate many different motor units that are required for an action, for a muscle contraction. Okay, so one of the words that we talked about in understanding synaptic integration is that a neuron sums or adds up all of the information that it's getting from different neurons. And so this is called summation. Okay, and there is spatial and temporal summation. This slide is about spatial summation. And this occurs due to the convergence of signals onto a single neuron. Okay, so in space, these neurons are converging on one neuron, which you can see that happening in the picture. Neurons, this neuron and this neuron are converging on this third neuron, and whatever EPSPs and IPSPs they are creating are then added together to determine the potential at the axon hillock. But then there's also the phenomenon of temporal summation, okay, and it says due to successive waves of neurotransmitter release that add together at the initial segment of the axon. So for example, in this picture, you have an EPSP, it didn't reach threshold, just got a little bump there, but yet if you have more than one EPSP in successive waves, they can sum together to create an action potential. Okay, so synaptic integration results from both spatial and temporal summation. Okay, and then synaptic integration could also result in inhibition of an action potential. And there are two types of synaptic inhibition: postsynaptic inhibition, which is produced by inhibitory neurotransmitters like we talked about, glycine and GABA, which are going to hyperpolarize the postsynaptic membrane, making it less likely to reach a threshold at the axon hillock. But also there's this phenomenon of presynaptic inhibition. Okay, in this case, an axon from one neuron synapses with the axon of a second neuron and sends gabatory or another inhibitory neurotransmitter to slow down the impulse before it reaches the terminal bouton, and then result in an inactivation of calcium channels. And one example that is talked about in our book is the example of how an opiate acts as an analgesic. Okay, so an analgesic is a drug that is going to act like a painkiller. Okay, so what's happening here, we see that opiates depolarize the axon, and that inactivates the calcium channel. And if that neuron was going to release substance P, remember that one involved in pain, then the opiate has now inhibited the release of substance P. That prevents a person from being in pain. Let's switch gears and talk about the topic of synaptic plasticity. And so synaptic plasticity refers to the ability of synapses to change in response to activity. So it has been found to be due to neurotransmitters, which is why it is part of this lecture. And it can also involve enlargement or shrinkage of dendritic spines, which is kind of seen in this picture, these outgrowths of the dendrite. Okay, so one phenomenon of synaptic plasticity is that the more you stimulate a neural pathway, the greater the excitement of that pathway. So we're talking about learning here. And so synaptic plasticity is really this phenomenon that says that you can change your brain. And it's, we used to think that children were born and their brains were growing, but that after childhood you really didn't have that many capabilities for learning. But now we think differently about the brain. And the brain's plasticity will continue through adult life. And so one thing that's going to happen is that the more you use a neural pathway, the more likely you are to be able to use it, which is called long-term potentiation, that leads to long-term memory. And so if repeated use increases synaptic transmission, then this is going to promote long-term potentiation. Repeated stimulation enhances or potentiates excitability. Okay, so in other words, if, the more you work with, say, flashcards, the more likely you are to be able to use those neurons that are about the recall of the terms on those flashcards. Okay, and one of the things that's been found to be due to is the insertion of AMPA receptors in the postsynaptic membrane. Okay, so one mechanism of learning that has been found to occur is that the frequent use of a neural pathway increases AMPA receptors, which leads to long-term potentiation. This event happens in the hippocampus, which is an area of the brain which has been found to be involved in making memories. And glutamate is the neurotransmitter that binds to the AMPA receptor. Let's take a look at how glutamate actually works. In this slide, what you can see is that what glutamate is doing is that it is going to bind to, I have NMDA receptor, but also AMPA receptor. The AMPA receptor and the NMDA receptor actually work together. And the binding to the NMDA receptor causes calcium to diffuse into the cell. And calcium goes through the NMDA receptor, which is a channel, and calcium binds to a protein called calmodulin, which then activates a second messenger system. In this case, calmodulin-dependent protein kinase initiates a variety of reactions that are involved in long-term potentiation. One of the things that happens is the increase in more AMPA receptors. Okay, so if you are working on your memory, repeatedly doing one activity, as in practicing the piano or practicing your physiology, you will actually build more AMPA receptors on the postsynaptic membrane. As well, what you can see in this slide is that nitric oxide can be stimulated as a retrograde neurotransmitter. It's going to go back to the presynaptic cell and increase the amount of glutamate that is coming out of the presynaptic cell. And so then this increase of glutamate from the presynaptic cell is then also going to stimulate a memory, or a long-term potentiation. Long-term potentiation also stimulates changes in the cellular composition. For example, it stimulates transcription and translation and the building of additional proteins that are going to cause these dendritic spines to form, and actually is going to change the structure of the neurons so that you can make more synaptic connections. And so studies of the NMDA receptor in rats, where they, enhanced the expression of additional NMDA receptors, showed that rats were smarter in studies where they're looking at the rat's ability to navigate a maze. And also, as I mentioned before, it's been found that AMPA receptors are increased in number due to long-term potentiation. The opposite phenomenon is this thing called long-term depression, if repeated use decreases synaptic transmission. okay, and repeated use decreases the excitability, reduces the efficacy of the synaptic connection, and this has been found to be associated with removal of the AMPA receptors from the postsynaptic membrane. Interestingly, this is sort of a homeostatic mechanism because if we continued to form more AMPA receptors and increased amounts of dendritic spines, that is actually sort of a positive feedback mechanism in which the brain could get overcrowded. So it is thought that long-term depression sort of counteracts long-term potentiation, and that this is also important for learning. Just as an example of long-term depression, endocannabinoids have been found to be involved in long-term depression by acting as retrograde neurotransmitters. And remember that retrograde neurotransmitters are going to suppress the release of a neurotransmitter from the presynaptic neurons. So this example, which is about depolarization-induced suppression of inhibition, which is kind of a mouthful, but depolarization of a neuron is then going to cause endocannabinoids to form, and then the endocannabinoids diffuse into the synaptic cleft. They're moving backwards into the presynaptic neuron and suppress the release of GABA. Okay, so GABA would be inhibitory, but now you've suppressed the inhibition, and so endocannabinoids can actually, in this way, be excitatory of long-term depression, and which would then lead to reduction of AMPA receptors and less long-term potentiation. But realize that both long-term potentiation and long-term depression depend on a rise in calcium ion concentration in the postsynaptic neuron. It's been found that a rapid rise in calcium ion concentration leads to long-term potentiation, but if there is very sort of slow, prolonged rise of calcium, this has been found to lead to long-term depression. And I think we will end our lecture at that point.